Hello everyone, I'm Boyang Liu from Shandong University. It is my pleasure to see you online. Today, we are going to study an important piece in reproductive medicine, fertilization and embryonic development. I'm not going to mention a lot of details, but just briefly introduce you basic issue in human reproduction. There are three main topics today: fertilization, embryo cleavage, and implantation. These are crucial steps in the first week of human development. During this week, an egg from the mother is matured and fertilized by a sperm to form a zygote. Then, the zygote undergoes cleavage to proliferate cells and becomes blastocyst. The connection between the embryo and the mother is established by implantation. So generally, these very first steps represent the beginning of a new life. In every month, around day 14 or day 15 of a woman's ovulation cycle, a secondary oocyte is matured by hormone regulation and turns into a metaphase II oocyte. This oocyte is then moved from ovary to the ampulla part of the fallopian tube, waiting for the arrival of sperms. After ejaculation, about 200 million sperms go into the vagina and up through the cervix of the uterus, and further up through the fallopian tube. The number of sperms starts from around 200 million to around 20 to 200 at the end of this long journey. So, only the fastest and most lucky sperms can meet the egg. 99.999% of the sperms lost their way reaching here. Fertilization is the first step of a life. When a sperm meets an oocyte, they fuse together and become a zygote. Within 24 hours of ovulation, the sperm needs to find the ovum in the ampulla part of fallopian tube. Before fertilization, sperms get the capacity from uterus and oviduct to be able to fertilize an oocyte. This process is called capacitation. Usually, around 20 to 200 sperms could arrive the ampulla, but only one could successfully fertilize. Surrounding the egg, there are corona radiata and zona pellucida to protect the female gamete. The sperm needs to penetrate these barriers to reach the egg. So, acrosome reaction takes place. When the sperm approaches the egg, the sperm is then activated and fused with the oocyte membrane. Many enzymes and lysosomes are involved in this process. Once the sperm is in, the properties of zona pellucida are changed and do not allow other sperms to penetrate, preventing polyspermy. Now, only one sperm successfully penetrates the zona pellucida and fuses with the egg. Its nucleus is released into the cytoplasm of the egg. There are 23 chromosomes in the sperm cell nucleus and 23 in the egg nucleus, meaning that both are haploid, namely pronuclei. Next, these two pronuclei form a diploid nucleus, and the ovum becomes a zygote. This one cell will eventually develop to an entire human body. By now, the fertilization process is complete, and the sex of the embryo is determined by X and Y chromosomes. This fused diploid nucleus starts its proliferation, and the zygote undergoes mitotic divisions. The cell number increases from 1 to 2, and then 4 cells, 8 cells, and more. These cells are known as blastomere, and this stage is called embryo cleavage. Blastomeres become smaller in each cleavage cycle, as the size of the embryo does not change, but the cell number is increasing. Early cleavage cycles are fast. In about 30 hours, the zygote turns into 2-cell stage, and in about 40 hours, it turns into 4-cell stage. At about day 3, there are 16 cells, and the embryo forms a structure called morula. At Morola stage, a process called compaction takes place, and the junctions between the cells are getting tight. Next, a cavity appears at the center of the embryos, and the Morola turns into blastocyst at day 5. There are 2 groups of cells in a blastocyst: inner cell mass and trophoblast. These cells start to differentiate to be more functional and specialized. In future development, inner cell mass will develop to the fetus, and trophoblast will develop to a part of the placenta. Early embryonic development, from zygote to early blastocyst, takes place in the ampulla of fallopian tube. At day 5 to 6, the embryo moves to the uterus and the zona pellucida is removed by blastocyst hatching. Now, the embryo is ready to establish a tight connection with the endometrium of the uterus. Therefore, a process called implantation takes place. When the embryo is ready, the mother also needs to prepare for the implantation. With the regulation of hormone, implantation window is open, and the endometrium is ready to receive the blastocyst to implant. There are roughly 3 steps to complete implantation: apposition, adhesion, and invasion. This is a precisely controlled process involving many signaling pathways. After these 3 steps, a connection between the embryo and the mother is well established. The embryo could receive oxygen and nutrients from the mother in order to grow. Okay, everyone. Today we have learned what happened in the first week of human development. This is a very important stage because any errors in this stage will lead to bad outcomes. Afterwards, the cells will become more differentiated and the embryo will further develop until an individual. Thank you very much for your attention.